Let's start with the main event. I'm Stephen Burley, Director of the RCSB Protein Data Bank and Founding Director of the Institute for Quantitative Biomedicine here at Rutgers, the State University of New Jersey. And the title of the presentation that Sagar, Khare, and I will be giving today is Computational Studies of Coronavirus Protein Structure Evolution in 3D. Here's an outline of the topics we'll cover, where the bullet motif changes to an arrow. Sagar will take over and describe some recent work that has been, some of which has been circulated as a bioarchive preprint. Other aspects of it are about to be submitted and posted on bioarchive. I'll talk about work that has already been published. So very briefly, the Protein Data Bank underpins all of this work. This was the first open access digital data resource in all of biology, founded in 1971 with just 7 protein structures. It's grown enormously now to more than 185,000 structures and has become the single global archive for protein and DNA and RNA experimental structures. Originally, this was a US-only effort, but in 2003, the Worldwide Protein Data Bank collaboration was established to include data centers in the US here at Rutgers and in Europe at EBI at Protein Data Bank in Europe, and in Asia, in Japan, at Protein Data Bank Japan. Subsequently, the WWPDB partnership was expanded to include EMDB and BMRB, both specialist data resources. And the images on the right show some of the structures that inspired the launch of the PDB as the first open access digital data resource in all of biology. Here at the US-funded RCSB Protein Data Bank, we convert global data into knowledge in a 4-stage process that involves deposition biocuration, which we do with our Worldwide Protein Data Bank colleagues, archive management and access. The RCSB PDB is responsible as the archive keeper for safeguarding the archive within the WWPDB partnership. And then we have both data exploration and outreach and education services that make these 185,000 plus structures directly available to more than 400 different scientific resources, available through our research-focused website, rcsb.org, to many millions of unique users annually, and through our education outreach website, pdb101.rcsb.org, to more than 850,000 unique users in 2021. Sorry, there's a typo at the bottom of this slide. The SARS-CoV-2 timeline is probably all too familiar to most, if not all of you. In late 2019, an unexplained influenza-like illness was seen in Wuhan in the People's Republic of China. In January of 2020, the causative agent was identified as a novel coronavirus, and its RNA genome sequence was released on the 10th of January. Come February 2020, PDB structures, SARS-CoV-2 structures began pouring into the Protein Data Bank, the first of which was that of NSP5, or the main protease, which will figure prominently in today's discussion. That's PDB ID 6 Lima uniform 7. And today there are approximately 1,750 COVID-19 related PDB structures in the archive. To the right, you can see David Goodsell's portrait of a respiratory droplet containing a SARS-CoV-2 virion. This is the unseen enemy, and this is why masking and social distancing is so important as non-pharmacologic public health measures. Our response at RCSB PDB was to make a host of information available to both expert and non-expert users. We've prioritized biocuration of COVID-19 structures, including getting them out the door very quickly, and doing any post-release revisions that might be necessary. We have strongly encouraged our depositors to release these structures immediately, and in virtually all cases, that has been the case. We're using consistent taxonomy and naming for the organism, for the virus, and consistent naming of the proteins. And we've created a coronavirus resources page, which you can access using this link. One of the most popular videos made by Maria Voigt, our graphic designer at RCSB PDB Rutgers, shows why hand washing is so important in combating the spread of the virus. A little about the organization of the genome before we talk about the evolution of the proteins in 3D. This is a single-stranded positive sense 5' capped 3' polyadenylated messenger RNA genome. Most of the non-structural proteins are expressed in a polyprotein and one of two polyproteins, which require cleavage by two viral enzymes, the PL protease that cuts the polyprotein at 3 sites, and the main or 3C-like protease NSP5, which cuts the polyprotein at a total of 11 sites. And then there's a series of subgenomic RNAs that get manufactured after the initial infection that encode the structural proteins, and you'll hear about spike extensively, and some additional non-structural proteins, which are the ORFs here. Returning to the very first SARS-CoV-2 PDB structure, that of the main protease, there are now approximately 430 crystal structures of this protein in the PDB with more than 300 distinct bound ligands. As I said, this is an essential enzyme responsible for cutting the viral polyprotein at 11 sites. And you can see it's a symmetric homodimer depicted here on the right. And there are 2 active sites, one here occupied by a covalently bound inhibitor on the front side, and then peeking out from the back side, you can see the other covalently bound inhibitor. This is a bona fide drug discovery target and is the target of nirmatrelvir from Pfizer that was recently, recently approved, and in combination with ritonavir, which is a cytochrome P450 inhibitor, and the combination of those 2 small molecule drugs reduces the likelihood of hospitalization in appropriately treated unvaccinated individuals by about 90%. It's phenomenally successful in interdicting the maturation of the viral infection by blocking the polyprotein processing early in the stage of early in the infection. So that's not, of course, the only structure of a SARS-CoV-2 protein we have. In fact, we've got 3D structural information for virtually all of the, either directly from the PDB in the guise of experimental structures, or in the form of computed structure models for nearly all of the proteins that are encoded by the SARS-CoV-2 genome. On the left, you can see an image here from a painting from David Goodsell showing SARS-CoV-2 fusing with the plasma membrane and ejecting the viral RNA into the cytoplasm. We used this information and access to this information from the PDB and from computed structure models of SARS-CoV-2 proteins to do a study which has, as I said, has now been published and had some very exciting outcomes. What we set out to do was to understand how each of 29 SARS-CoV-2 study proteins changed in amino acid sequence and changed in 3D structure during the first 6 months of the pandemic. In all, we analyzed the proteomes of about 49,000 different viruses. We found that every one of the 29 study proteins had undergone some mutation in some of the different viruses that were isolated from infected individuals during the first 6 months of the pandemic. We looked at more than 1 million clean viral protein sequences and found that nearly 1.1 million of these, 88%, were actually unchanged across the study proteins. The lowest level of conservation we were able to detect was 58%, and the highest level of conservation was 98%. On average, we saw one unique sequence variant for every residue in each study protein. So if a study protein was 100 amino acids in length, we would typically see 100 distinct unique sequences among the 49,000 proteomes that we analyzed. So mutations, substitutions of amino acids, missense changes in the gene are actually quite rare. Most of the substitutions we found were single substitutions. That was 2/3 of the unique sequence variants. There were doubles, triples, quadruples, even quintuples and more. About 1/3 of the changes, that the substitutions that we saw, were conservative versus 2/3 being non-conservative, and we didn't consider either as insertions or deletions. Most of the mutations arise from single base changes. You'll see in a moment that we were able to map the locations of all of the amino acid substitutions to different parts of the protein, including the entire set, either the surface amino acids, the hydrophobic core amino acids, or those involved, or those in between the surface and the core in what we call the boundary layer. These tables show the frequency of a reference sequence, a reference amino acid becoming a substituted residue of a different type. And if we look in more detail at this grid for the core amino acids with reference to the genetic code wheel here on the right, we can see that many of the substitutions that were observed in the first in this 29 study proteins during the first 6 months of the pandemic actually were the result of single nucleotide changes. So for example, here, a tyrosine in the reference sequence becoming a cysteine is, let me just make that a little bit smaller so I can see more of my screen. So we've got the codons, the possible codons for tyrosine here, and the possible codons for cysteine here. And you can see that if an A is changed to a G in the middle of the codon, then you go from a tyrosine to a cysteine, and the same is true for these other examples that I flagged with arrows. Not surprisingly, as we've grouped the hydrophobic amino acids together in the first part of the column and the first part of the row here, so you can see that there's a lot of hydrophobic turns into hydrophobic, but that's not exclusively the case. Sometimes hydrophobics turn into polars, and sometimes polars turn into hydrophobics. So moving on to the results of the structural modeling, we made computed structure models, generated computed structure models for every one of the unique sequence variants of every one of the 29 study proteins, and these have all been made publicly available. Only a minority, 7% of all substitutions, map to the hydrophobic core, the remaining being split equally between the boundary layer between the core and the surface of the protein itself. This is not surprising because hydrophobic cores are generally intolerant to substitutions. And then the Rosetta tool was used to actually understand the energetics of the changes that we saw in terms of the substitutions. And the summaries provided here on the right, nearly 3/4 of the substitutions were mildly destabilizing in terms of the calculated free energy change, comparing the original reference sequence to the substituted unique sequence variant. Approximately 1/4 of the substitutions were stabilizing, meaning that the computed delta delta G was negative, and approximately 5% of the substitutions yielded results that we deemed to be unreliable in terms of the calculated energy change, and we didn't consider that minority of cases further. These are outliers that are probably reflect some weaknesses in the method that was used to actually understand the energetics. So the way the computational modeling of the unique sequence variants was done is depicted schematically here. We took the PDB structure, or the computed structure model, from homology modeling, energy minimized it, and then did repacking minimization to generate the reference model, and then the same for the unique sequence variant. Using the Rosetta physical statistical potential, which takes into account things like van der Waals, electrostatics, solvation, conformation, and pair potential, and other terms, including the Ramachandran diagram, it was possible to actually compute the Gibbs free energy change for comparing folded versus unfolded amino acid sequences. And then we computing a delta G for the reference and a delta G for the substituted, we're able to compute a delta G that reflects the impact of the single amino acid, single or double or triple amino acid changes. And when we actually depict the distribution of these computed delta Gs, we see a distribution that is skewed to the right towards the unfavorable, the destabilizing changes, and we were able to further break this down into two Gaussians, one centered very close to zero, and one centered at a higher unfavorable change in the energetics of protein structure stabilization. Interestingly, very similar distributions have been observed by others doing both computational and experimental studies of the impact of single amino acid substitutions in globular proteins. So the calculation is reproducing what is seen in experiment. If we look further and consider the different types of substitutions, surface, boundary, and core, we're able to subdivide this energy histogram and see that in terms of the surface changes, that's the Gaussian for the distribution of those energy changes is centered quite close to zero, to neutral. Boundary, it goes a little bit more unfavorable, and then when you go to core, it goes to even more unfavorable, to +5 Rosetta energy units, which is nominally 5 kcals per mole destabilization. So this is not surprising at all. In fact, when substitutions occur in the hydrophobic cores of proteins, they are generally destabilizing. And this is a result of perturbation of the close packing of amino acid side chains in the hydrophobic core of the protein. So the key lessons learned from this study were, one, that substitutions are relatively rare. And the reason for that is that the enzyme encodes an NSP14 proofreading, the virus encodes an NSP14 proofreading enzyme. Many of the non-conservative substitutions we saw result from single base changes. Only a minority of the substitutions occur in the hydrophobic core. The distribution of the stability effects follows a bi-Gaussian pattern that's been previously seen for globular proteins. But importantly, when we analyze these structures in detail, the key enzyme active site amino acids in the proteases, in the proofreading enzyme, in the RNA-dependent RNA polymerase were rarely, if ever, substituted, making these proteins attractive targets for structure-guided drug discovery. And we believe that modeling of the substitutions in 3D and making this information available publicly may aid in prospective identification of potential drug resistance mutations. This work was done primarily by a dozen 2020 summer interns in the Rutgers RISE program, the Research Intensive Summer Experience. Their smiling faces are depicted here, and their names are listed in the column on the right. Anyone in the audience coming in from outside Rutgers who's interested in the RISE program can find more information here. You need to be applying, you need to have actually applied probably in the fall for summer of 2022. I don't know if the applications have closed, but please investigate if you're interested. We published this work, including very detailed analyses of what happened to all 29 of the study proteins in Proteins, in the journal Proteins, in a special online COVID issue. All of the 12 RISE students, plus all of the Rutgers Institute for Quantitative Biomedicine summer 2020 bootcamp instructors and participants were listed as co-authors. The method for actually analyzing the structures and pursuing this study on all 29 proteins was worked out at the bootcamp with all of the individuals listed in the on the face page of the paper here. 64 in total, underscoring the reality that today research is a team sport, and understanding how to work well and effectively and synergistically with others is going to be central to your success as early career scientists. So please remember, research is a team sport. I introduced NSP5, the main protease, very early in the talk for a reason. It's going to figure prominently in Sagar's part of the presentation. But I also want to reiterate a point I've made earlier in this same format. The main protease is very, very highly conserved across all of the coronaviruses, and you'll see that in more detail. If we compare SARS-CoV-2 with SARS-CoV, the main protease amino acid sequences are 95% identical. If we look at the active sites, meaning the amino acid side chains that are lining the binding site for this inhibitor here, the location of one of the two active sites, the other being over here in this overlay of the two proteins, you can see, I can tell you that the active site non-hydrogen atom root mean square deviation is less than half an angstrom. So the differences, the structural differences between the SARS-CoV-2 active site and the SARS-CoV active site is less than, on average, less than half the diameter of the hydrogen atom. And what this tells me as an experienced drug hunter, having spent a decade in the pharmaceutical industry, is that a safe and effective drug against SARS-CoV main protease would have worked for SARS-CoV-2. And so I think it is a sobering lesson for all of us that investment of a few $100 million back in the 2000s could have prevented millions of deaths and literally tens of trillions of dollars of economic losses. Now with this, I'm going to stop sharing and turn over to Sagar, who will reshare the same presentation and take you through the results of work that has either only been reported as a preprint or has yet to be publicly described. So thank you very much for your attention, and thank you, Sagar, for being such a terrific collaborator and Institute for Quantitative Biomedicine citizen.
Thank you, Stephen. So let's see, I'm going to share my screen and all right. Let me move this stuff away a little bit. Okay, so so as Stephen described, we developed this method in looking at the impact of amino acid substitutions as the SARS-CoV-2 virus was traveling all over the world, keeping at the back of our mind, the lesson that Stephen ended with, which is that, looking at a conserved target such as main protease would be fruitful. And indeed, as shown by Pfizer, they have come up with a really effective drug against SARS-CoV-2 by developing inhibitors against the main protease. So the question on our mind now is sort of twofold, right? One is, how effective is this drug going to be? And can we anticipate the evolution of potential drug resistance? So for the second part, some of the methods that we have developed, should be applicable, and we're indeed collaborating with experimentalists to try and prospectively predict drug resistance mutations. But for the first part, it's a little bit more challenging, right? How can we be prepared for something that we don't know yet or haven't identified yet? So I'll take you through our thinking on that and where we're headed. So first, just as a quick recap, this NSP5 main protease is involved in coronaviral maturation by making very specific cleavages at 11 sites in the proteome, right? So it's also equally important to recognize that it is not cleaving elsewhere, right? And all of these 11 sites are somewhat similar, as I'll show you, but they are still different, right? Which means that if you think about the pressure on this protease, it has to be able to maintain its cleavage, even if it's changing somewhat, right? Or the cleavage sites all have to simultaneously change, which can be far more unlikely in the course of natural evolution. So with this in mind, the idea that we had for thinking about how we can be prepared was was inspired by looking at the diversity of the proteases in the beta coronaviral sarbecoviral lineage that have been characterized. So for example, here, as we have just seen, there is a very high degree of similarity between SARS-CoV-1 and SARS-CoV-2. And the first structure, in fact, of SARS-CoV-1 was determined by Stephen's group at SGX Pharmaceuticals and published in the New York Times rather than a scientific journal. I wish that we would be able to publish something in the New York Times. But anyway, so the idea is, what if we look at the diversity of main protease sequences across coronaviruses, right? So there have been many coronaviruses that have been identified and continue to be identified. And a fact about coronaviral evolution is that recombination plays a huge role. So people have recently sequenced coronaviruses from bat species across Southeast Asia. And what they have found is that these coronaviruses often coexist. So coronaviruses from different lineages like alpha and beta and so on can coexist within a single organism, and they are constantly recombining. So if we want to anticipate the evolution, then we really have to think about the broad diversity of coronaviruses. And so here's a phylogenetic tree of all coronaviruses, coronaviral main proteases, according to sequence. And you can see that they cluster nicely with beta coronaviruses, including the ones that we have seen, such as SARS-1, MERS, and SARS-CoV-2. They cluster quite closely. As you can imagine, as you start drifting away from SARS and SARS-CoV-2, the active sites are likely to be different. So our approach that we have been developing is to broadly survey these coronaviral active sites, not only in sequence, but also in terms of three-dimensional structure, and identify such patterns here that are shown on the right. So this is highly conserved between SARS-CoV and SARS-CoV-2, but things are somewhat different for MERS and are likely to be even more different for all these other coronaviruses whose structures have not been yet experimentally determined. So in fact, what we can do is we can create 3D structural models using the recent revolutionary advances in protein structure prediction, such as the development of AlphaFold and Rosetta Fold, and compute these three-dimensional models with a high degree of confidence, especially because they are so similar in some sense to other proteins whose structures have been determined, and we can broadly survey the diversity. So here is all of the active site positions whose numbers are shown in the bottom, and you can see the amino acid diversity shown here as a logo plot where the size of the letter essentially tells you how conserved it is. For example, at site 172, which is a catalytic site, there is 100% histidine, as you can imagine. And site 145, which is the catalytic cysteine, is almost 100% cysteine as well. So the idea is that by looking at structure, we can get a sense for what the diversity of this active site is going to be. And that is going to tell us how effective countermeasures such as drugs that we develop against main protease are likely to be effective against coronaviruses that have yet to jump the species barrier. A different way of looking at that is to also look at these 11 sites that this main protease cuts, right? Because this is an important functional restraint on the evolution of this main protease. So looking at these sites also gives us a sense for what is the evolutionary space that is available for the main protease sequence to drift. And again, you can see hearteningly that there is a high degree of conservation. So for example, the bond that is cut is between this P1 and P1 prime residues, and P1 is almost exclusively 100% of the time Q or glutamine, and a small amino acid such as serine or alanine at the P1 prime position. So this tells us that there is a high degree of selection pressure on the main protease in order to maintain its function. But nonetheless, there is some diversity here. And as you can imagine, small molecule binding can be abrogated or ablated by really small changes in the active site. So having these three-dimensional structural models provides us with insights into what kind of changes may therefore affect drug binding. So one thing that is important to notice is that the phylogenetic tree that we can construct based on sequences can be quite different compared to the phylogenetic tree. The details of the phylogenetic tree can be quite different based on the three-dimensional structure. And in fact, even though you see this kind of conservation here, we can see that there are differences, significant differences in other coronaviruses. And so what we have done is identified, based on clustering, three-dimensional clustering, identified representatives of different branches of this phylogenetic tree. And we are now starting to study these both in the lab as well as computationally to try and examine how their substrate specificities are different, and whether the inhibitors that have been developed are likely to be effective against these, right? And so as a preliminary example, you can see a superposition of the models of these 5 representative proteases on the SARS-CoV-2 structure bound to the Pfizer drug, nirmatrelvir. And you can see that the drug is predicted to at least fit quite nicely in the pockets of these other proteases as well. Of course, it's still an open question about what happens if you start to make mutations in these, and if they start to drift as well, how effective this drug is going to be. So our hope is that by looking broadly at this diversity of coronaviral proteases, as well as looking locally around each of these nodes that we have identified for how the viral sequence is going to drift around these nodes, we might be able to say something more quantitatively about the effectiveness of inhibition of this particular drug, and therefore the efficacy of the drug going forward and aiding pandemic preparedness in that way. So these results are currently being written up. But while we have been looking at this around Thanksgiving, we all heard about this new variant, which certainly to me increased my knowledge of the Greek alphabet a little bit. So this Omicron variant came about and has swept the world. And there are many substitutions, right? So we heard about how many mutations there are. And in particular, a lot of these mutations, about 15 of them, are in a very small region of the spike protein of the virus, right? So at this point, I think all of us know what the spike protein is. So I'm not going to introduce that protein, but suffice it to say that this is the protein that is involved in binding to the ACE2 receptor on human cells, and as well as quite importantly, antibodies that have been generated as a result of vaccination or as a result of therapeutic development target this particular protein, and in particular, many of these antibodies target the receptor binding domain. So what is going on with Omicron, and what can our models tell us about what to expect, as well as perhaps prepare for Omicron as well as other strains that may arise in the future. So here, what we did with graduate student Joey Lubin and graduate student Chris Markosian from Wadi Arap and Renata Pascolini lab, in collaboration with Stephen and me, and Bala Desingu from the Office of Advanced Research Computing at Rutgers, we started investigating what this Omicron RBD, this particular part of the protein, the business end, so to speak, of the spike protein is going to look like. And so here you see the sites of changes in Omicron highlighted on the RBD structure. This was determined for the Wuhan strain. And in gray, you will see this ACE2 binding region, right? So a lot of these changes are occurring in the ACE2 binding region, and some of them are also occurring in a region that is somewhat distal from ACE2 binding. So there are obviously several questions that arise. Clearly, the virus should be able to maintain ACE2 binding, but how does it affect ACE2 binding? And how does it affect the various antibodies that have been developed or that have arisen to recognize these particular regions of the virus? So those were the questions that we asked. And the way we did that was similar to the approach that Stephen described that we developed for our overall SARS-CoV-2 evolution study, except that now we also looked at antibodies. So it was not just looking at proteins by themselves, or the spike protein by itself, which we had done, but now we're also looking at complexes with ACE2 and the various antibodies that are known to recognize and whose structures are widely available, are available in the PDB. So we start from the PDB structure, we create an antibody model or an ACE2 model and perform the same kind of calculation, except that in this case now, we allow a little bit of flexibility in the modeling. And so in this case, we're going to compare the reference with the Omicron sequence and allow some flexibility for both the reference and the Omicron. And that gives us two numbers, these delta Gs, not of stability now or folding now, but of binding. And while we were doing this, of course, the structure of the Omicron spike protein and RBD was not known. And so we utilized AlphaFold to develop a second pipeline, right, that we combined with the first one, where instead of using the PDB structure, we were using the AlphaFold based structure prediction. And similarly, we did the wild type and the Omicron, and we generated using a similar pipeline. We were able to generate two more numbers corresponding to every complex, right? So the basic take home is for every complex with ACE2 or with one of the almost close to 300 antibodies or other binding domains, we were able to get 4 numbers for each and then derive a kind of consensus prediction based on these modeling approaches. So with that in hand, let's look at what our models are predicting and how that compares to the experimental results that have since been determined. We posted this manuscript on the 13th of December, and almost about a month later or 3 or 4 weeks later, structures started to come in. So we were able to benchmark our approach. So here's the ACE2 binding domain complexed with the Wuhan HU1 or the original pandemic strain. And you can see that there are certain interactions that are being made across the interface, a few hydrogen bonds and electrostatic interactions. And now, if we look at the same region now, right, we can divide it into roughly sort of 2 parts, right? So one is the region on the left, which is spike RBD residues in the 490s, 500s, and the other region was on the right? So let's look at this region, how that has changed when the spike has come in, when the Omicron mutations have come in. And what you can see clearly is that the multiple mutations in Omicron are sort of playing together, right? Or in the language of genetics, this is called positive epistasis, or in the language of biochemistry, positive cooperativity, where extensive network of hydrogen bonds and electrostatic interactions are being made according to our models, as well as some new interactions are being generated by sites of mutations, which are highlighted in red. So the first thing that we noticed was that there appears to be a lot of cooperation, suggesting that Omicron is the result of a long evolutionary process where each of these individual mutations may not do very much or may increase binding affinity, but when they all come together, that certainly leads to a very viable and robust binding interface between Omicron spike and ACE2. In fact, so this is our computed structure model that was released on 13th of December, and then 2 weeks later, the experimental structure was released. There are sites of close agreement between our model and the computer structure. Of course, we didn't get everything 100% right in all of our models, but in terms of the consensus, we got most of the interactions that were then reported in the experimental structure, which is of course also an average of various conformation. So we were heartened to see that our modeling was able to recapitulate experiments largely and then use that, and that gave us confidence in trying to understand how it may affect natural and engineered antibody binding, for which currently no structures are known. So if you think about the antibodies that have been generated against the RBD, they correspond to 4 classes, right? So in terms of a football analogy, you can think of them as the defense team that can attack the quarterback or the running back from many different directions, right? So that's exactly what our immune system does. And it generates these 4 classes of antibodies that bind to different parts of the structure. So class 1 and 2 neutralizing antibodies, they can be generated as a result of vaccination or as therapeutic development, bind to the same receptor binding motif, which interacts with ACE2. However, there are other classes of antibodies, for example, S309, which binds to, which sort of binds side-on, and class 4, such as CR3022, which binds on the other side, right? So our immune system, as well as therapeutic development, has resulted in a sort of broad recognition of the spike. And so the question is, what's happening with these various classes of antibodies? And there are, as I said, around 300 structures known. So we used our pipeline to generate models of the Omicron RBD bound to all 300 known or previously determined complexes of RBD with the spike protein. And you can see the summary of our results on the right here, right? So here are the 4 classes. This is the number of antibodies that were considered in every class. And here are the 15 listed mutations that, that are sites of mutations are highlighted in gold here on the RBD structure. More red a cell is in this table, the more destabilizing this mutation is, according to our calculation, a consensus calculation for all of these antibodies. And so one of the things that jumps out is that, it may or may not be very clear here, but there's slight sort of red everywhere. So all of the mutations are generally destabilizing, except a few notable exceptions. But there are a few mutations that are so strongly destabilizing for particular classes of antibodies, in particular class 1 and class 2 antibodies, they are totally destabilized. And indeed, these therapeutic antibodies that were developed against class 1 and class 2 were found to be, totally, not able to neutralize Omicron anymore, which probably explains the breakthrough infections that we see, as well as, that prompted the FDA to discontinue the EUA that was given to some of these antibodies, which targeted this region, right? On the other hand, class 3 antibodies seem to be, an example here is this antibody S309, which is the base antibody in Vir Biotechnology and GSK's antibody that has maintained approval because it does appear to be effective. Its binding is not really very effective, and our models show that there is a little bit of destabilization and a little bit of stabilization. So this binding is not expected to be affected very much, and indeed that has also turned out to be the case in experiments. So our calculations, were able to sort of predict in some sense what was going to happen to the antibody response. And in fact, we can take a look at the structural basis for why this weakening happened, right? So let's look at a few examples. So the first one we can think of is this class 1 antibody, right? Where this K417, which is a site of Omicron substitution going from K to N, was involved in this beautiful, tridentate sort of, cluster of interactions, both salt bridges as well as hydrogen bonds. And that has totally disappeared, right? So you can see that this has created an empty space. This N asparagine in the Omicron RBD much prefers to make intramolecular interactions as opposed to intermolecular interactions, and there's just not enough space. And so you can see why this might be destabilized, creating a big hole that would be extremely destabilizing for binding. Of course, it's not always the case that everything is destabilized. Sometimes you can get sort of, glancing interactions that can be maintained. So for example, here, you can, you lose one interaction in going from Y to H here at 505, this backbone hydrogen bond. But on the other hand, you can get a compensatory hydrogen bond here perhaps, and these type of glancing interactions in going from Q to R, right? So that explains why sometimes we can get a little bit of stabilization. Of course, if this, if the interaction shown on the previous slide was far more dominant, these interactions would never come to be realized, and that may, so these may be a little bit moot in that sense. So we have to look at these data as a whole, and therefore predict that class 1 antibody interfaces are significantly destabilized, right? So we can again look at a class 2 interface, again going from this K417 to N417, and how this beautiful, interaction network is totally destabilized here, again explaining why class 2 interfaces are destabilized. And another class 2 interface, another sort of mutation that has been key, is this E484 to A, and again you can see both a salt bridge and a hydrogen bond interaction are totally disrupted. So that's well and good, but can we do something more with this, right? So we have, what we have is an atomic resolution 3D model of how these interfaces have been, altered by these Omicron substitutions. So it stands to reason that we should therefore be able to change these antibody interfaces, perhaps by redesigning the antibody to try and make more interactions that overcome the destabilization, right? So it's not immediately clear how you would do it in this case, perhaps putting in some hydrophobic residues that fill up this space, but there are other opportunities. And so we decided to systematically investigate how we can redesign these antibodies to perhaps now start being more effective, right? And in general, the pipeline that we envision developing in future work is to prospectively generate mutations on the RBD, and then prospectively generate antibodies, that can overcome the loss of binding that can be introduced by these mutations that are both seen and unseen yet. So, okay, so this is what we did, and so we re-engineered computationally, at least for now, a Regeneron antibody, which is a class 1 antibody, and as expected, this when this T478 became a K, the backbone hydrogen bonding interaction was lost. However, by making 2 compensatory changes in the antibody, right? So for example, in this case, we can change the antibody N93 on the antibody to a D, and introduce a histidine to reintroduce a hydrogen bonding network, which should be favorable for binding, right? And similarly, you can see here that in going from Q to R, in this case, there was no interaction that was lost, but this R can be targeted by making further targeted substitutions on the antibody to make interactions. So in one case, we can overcome interactions that have been lost. In the other case, we can take advantage of the mutation, right? So we're sort of trying to match the virus by changing the antibody as well. And importantly, computation is much more fast compared to experimental validation. So this allows us to generate many hypotheses that then we are hoping to test broadly in developing a pipeline for antibody redesign, so that we can be prepared for any further changes in the virus, right? So to summarize, I told you about 2 stories in which we are thinking about how to anticipate the changes in the virus. In the first one, I told you that the coronavirus main protease active site and its cleavage sites are highly conserved. And it's possible, given this conservation, that inhibitors that have been developed can act as near pan-coronaviral inhibitors that may counter strains that we have yet to see, as well as provide us information about resistance mutations that may arise. And I told you about what Omicron mutations and protein substitutions are doing, and how this can lead to re-engineering therapeutic antibodies, not only against Omicron, but against potential strains that have yet to arise. And so in summary, modeling of coronaviral proteins in these 3 case studies that we described to you, made in the structure-based drug discovery of broad spectrum pan-coronaviral inhibitors, may allow prospective identification of drug-resistant substitutions that may ablate the binding of these inhibitors, and may enable the engineering of therapeutic antibodies for not only existing variants, but also preparing for variants that have yet to arise. So a lot of this work over the years has been spearheaded by Joey Lubin in my group, who really has worked with all undergraduate students as well as other collaborators, and everyone in the lab has pitched in. And please check out our lab website. We work in protein design and engineering, and are excited to hire new people who will be working on this COVID and other projects. And also thanks to other people who helped out. And thank you for your attention.